Assalamu alaikum. I'm Insha Majid, MPhil student from Department of Botany, University of the Punjab, Lahore, and the topic of my presentation today is microspore analysis of some members of the family Brassicaceae from Lahore, Pakistan. Here are given the contents of my presentation. Basically, palynology is the study of the palynomorphs, which range in size from 5 to 500 micrometer in both the living and fossilized form. And pollen grain is a container which houses the male gametophytic generation in both the angiosperms and gymnosperms. Palynology have various applications such as plant taxonomy, forensic sciences, aeropalynology, food quality control, systematics, and phylogeny. Lindley was the first person who used the pollen characters in the classification of the family Orchidaceae, and now the several workers has worked upon the pollen morphology of the plants related to the different families. Palynology was realized for the very first time, and it was recognized as a separate section of International Botanical Congress in Sweden in 1950. Carolus Linnaeus for the very first time considered the family Brassicaceae as a natural group, and he named it as Tetradynamia. And in 1948, John Hutchinson suggested that Brassicaceae is derived from the Papaver, and it was diverged approximately 140 million years ago. Brassicaceae is a cosmopolitan family, and it is worldwide in its distribution. But the members are mainly found in the temperate zones and especially in the Mediterranean regions. In Pakistan, it is represented by 92 genera and 250 species. Pollen morphology of the family Brassicaceae has been examined by the several workers, but there are no reports on the pollen morphology of the family from Pakistan, or there are only one or two reports on the pollen morphology of the family. But on the no work has been done on the pollen morphology of the family from Lahore, Pakistan. Basic aim of this study was the collection and the preservation of the flowers, so that the pollen morphographic characters of the different collected plant species can be examined, and the data obtained from the examination of the pollen morphology of the different plant species can be used to construct the dichotomous keys, and it can be used to assess the classification and taxonomy of the plants. A field survey was carried out for the collections of the flowers from the different localities of the Lahore. Basically, the species were collected from the botanical garden and from the vegetable farm of the Punjab University, Lahore, Pakistan. Other than this, different nurseries such as Changa Nursery and Abbas Nursery were also visited, and Jinnah Park was also visited for the collection of the flowers. Study area basically included the city Lahore, and the map of the Lahore city is shown here, and the basic area of the study was the Punjab University, Lahore. In this table, location and collection date of the 8 plant species of the family Brassicaceae from Lahore, Pakistan has been given. This table represents the habitat and climatic zone of the collected species from Lahore, Pakistan, and it can be easily seen here that all the collected species were herbaceous in nature, and their habitat were cultivated fields because most of them are the food crops. But the Iberis amara was collected from the dry and open areas, and it was also collected from the gardens because it is an ornamental plant. Research methodology which was carried out included the separation of the anthers, and then the fixation was performed. Fixative contained the glacial acetic acid, ethanol, and formaldehyde. Maceration of the anthers was performed by carrying out the decantations in the centrifuge tubes, and the alkali treatment was carried out with the potassium hydroxide. Then again the neutralization with water was carried out, and the sample was preserved in the 1% thiomersal solution. Staining of the sample was performed in the safranine, and mounting of the sample was performed on the clean glass slides, and glass slides and cover slips were cleaned with the ethanol, and microphotography was performed with the trinuclear microscope, MIGI trinuclear microscope at the high resolution power, that is X60 and X40. This picture here shows the diagrammatic representation of the methodology which was carried out, including the separation of the anthers, maceration, their fixation in the centrifuge machine, and then the staining, further the mounting, and then further analyzing them with the help of a trinuclear microscope. Results and discussion are further discussed. This table shows the pollen type of the 8 collected species of the family Brassicaceae from Lahore, Pakistan, and it can be easily seen that Only single pollen, that is Iruka sativa pollen, were colporate in nature, and rest of the pollen were colporate in nature. And this figure here shows the percentage occurrence of the pollen type of the collected plant species of the family Brassicaceae, according to which tricolporate pollen type was shown by only the 12% species, and tricolporate pollen type was shown by the 88% species. Here we have given the detailed pollen morphology of the collected samples of the family Brassicaceae from Lahore, Pakistan. Their pollen type, their pollen shape, pollen size, and exine ornamentation has been given in the table. This table shows the pollen character variation in the 4 collected varieties of the Brassica oleracea. 4 different varieties were collected, and their pollen morphographic characters variation has been shown here. Their pore size, their exine, and their pollen shape. It can be easily seen here that they were variable in their pore size, in their exine thickness, and in their pollen shape. This figure shows here the pore size of the collected species of the family Brassicaceae, and it can be seen here the maximum pore size was shown by the Brassica campestris, and it was 8 micron meter. The next figure shows the colpi width of the collected species, and it can be easily shown that the maximum colpus width was shown by the Iruka sativa species, and it was 12 micron meter. Here the colpi length of the collected species of the family has been shown here, and the maximum colpus length is shown by the Brassica campestris, which is 30 micron meter. Further, the pollen diameter of the collected species is shown, and the maximum pollen diameter is shown by the Brassica campestris, which is 28 micron meter. Further, the floral and pollen morphology of the plant species Iberis amara has been shown in the figure, and the tricolporate pollen grain is observed. Overall shape is subcircular to broadly triangular in nature, and the exine ornamentation is perforate to fovulate. The next figure shows here the floral and pollen morphology of the Brassica rapa, and it can be easily seen that pollen grain is tricolporate. Spheroidal shape is observed in polar view, and sub-oval shape is observed in the equatorial view. Distinct colpi are noticed, and the exine thickness is varied from the reticulate to vermiculate to punctuate in nature. Further, the pollen morphology of the floral and pollen morphology of the Iruka sativa species has been shown, and it can be easily seen that the tricolpate pollen is observed. Overall shape is observed to be varying from the subcircular to slightly lobate, and in many specimens intine was also visible. Distinct colpi are noticed, and exine is observed to be inarticulate to perforate with 1 micrometer thickness. Further, the floral and pollen morphology of the Brassica campestris is shown in the figures, and tricolporate pollen are observed. Overall shape is varying from the subcircular to lobate in nature, and distinct colpi are noticed, and pollen diameter is 6 to 8 micron meter. Exine ornamentation is varying from reticulate to perforate. Further, the floral and pollen morphology of the Brassica oleracea variety Sabelica is shown, and it can be easily seen that tricolporate pollen are observed, spheroidal to subtriangular or lobate pollen are observed, and distinct colpi are noticed. Exine thickness is silate to infrareticulate. Further, the floral and pollen morphology of the Brassica oleracea varieties acephala has been shown at power 60, and tricolporate pollen grain is recorded. Shape of pollen is subcircular to lobate, and distinct colpi are noticed. Further, the floral and pollen morphology of the Brassica oleracea varieties italica has been shown. Tricolporate pollen are observed, distinct colpi are noticed. The shape is spheroidal to lobate, and pollen diameter is 2 micron meter. Further, the floral and pollen morphology of the Brassica oleracea varieties botrytis has been shown, and tricolporate pollen are observed. Subcircular shape is observed in polar view. Further, we will go towards the conclusion. Microspore analysis of the 3 genera of the family Brassicaceae from Lahore was performed, and from these 5 plant species belonging to 3 genera, that is Brassica, Iruka, genus Iberis, and 4 subspecies belonging to the species Brassica oleracea were herbaceous in nature. 90% of the collected species were native, and only 10% were cultivated, that is Iberis amara. Two pollen types were basically observed, that is tricolpate and tricolporate. Tricolpate pollen type were observed in only one species of the genus Iruka mill, that is Iruka sativa, and tricolporate pollen type were observed in the one species of the genus Ibarus and three species of the genus Brassica and three subspecies of the Brassica oleracea species. Largest pollen were observed in the Brassica compactus with the pollen diameter of 28 micron meter, and highest corpus length was measured in the Brassica compactus, that is 30 micron meter, and lowest was observed with the Iruka sativa, that is 12 micron meter. And important pollen morphographic characters are pollen types, exine thickness, width and length of pulpi, pollen diameter, ornamental patterns, and these characters can be used to construct the identification keys for the classification of the plants. Here are given the references. Thank you so much for your time.